Hey, my name is Scott, and this is Kristen, and we're here to do our Week 9 genetics problems and explain how we got the answers to the questions that were asked.
Yep. Okay, so for question number 1, it asks us to diagram the intermediate stage of DNA replication. And so here's my drawing, and it says to make sure to indicate the polarity. So we have the 3' and the 5' ends on either side, and the OREC is here, and we have the DNA primase on there. Just going over here, and then helicase, which is also going to be over here, and then it says RNA primers, which are going to be in the red over here, which will help create a RNA strand for the Okazaki fragments to synthesize the lagging strand, and also there's one primer for the leading strand, and then DNA polymerase is down here, which is going to synthesize the DNA and take out the RNA primers, and then DNA polymerase 3 is here, which is going to synthesize the DNA, and then the Okazaki fragments are just the fragments next to the red primers.
Yep. Cool. So that was question 1. Question 2 here is mostly vocabulary. It's asking about some different definitions, and it asks us to match that up with what the word it's defining is. So the first one, it asks about a protein complex that synthesizes primers in eukaryotes, and the only one that synthesizes primers is primase, has it in its name. DNA A, that is a protein that promotes the separation of AT-rich sequences in the OREC, and then the subunit of DNA, that polymerase that contains proofreading activity, is the epsilon unit subunits. That helps with proofreading. DNA polymerase 3, the alpha subunit of it, it synthesizes the leading strand in eukaryotes, and so it works on the leading strand. TUS, it's a protein that displaces DNA polymerase 3 from the chromosome to terminate replication. DNA polymerase 1, it will go 5' to 3' in primase and in exonuclease activity. And then topoisomerase 2 is a protein that introduces negative supercoiling into the DNA replication fork. ORC is a protein that binds directly to the ARS elements. The single-stranded binding proteins, those are proteins that prevent DNA strands from annealing during replication. And then DNA ligase is an enzyme that seals Okazaki fragments into a continuous strand of DNA. And so it smooths out the DNA by putting everything together.
Yep. Okay, so for question 3, part A, it says to list the 5 enzymes that consume ATP during DNA replication. So we're going to have DNA A, DNA ligase, the MCM helicase, DNA helicase, DNA primase, and DNA gyrase. And for part B, it says list 5 enzymes that consume D-ATP during DNA replication. And that's going to be DNA polymerase 3, DNA polymerase 1, DNA polymerase delta, DNA polymerase alpha, and DNA polymerase epsilon. And for part C, it says list 5 enzymes that have 3' to 5' exonuclease activity. And that's going to be all 3 of the DNA polymerases, so 1, 2, 3, and then DNA polymerase epsilon and DNA polymerase delta. It says indicate one enzyme involved in replication that uses DNA as a template to synthesize RNA, and DNA primase does that, puts down the primers, and then for E, it says indicate one enzyme that uses DNA as a template to synthesize DNA, and we learned about that with telomerase.
Cool. So question 4, it gives us a sequence of DNA, and then it also gives us the primer, and it asks us to find the nucleotides in a reaction when we have certain kinds of, I forget what these are called, the ATP similar units. I forget what they're actually called. So the first one contains all 4 of the ones needed to synthesize a DNA strand, which is DATP, DCTP, DGTP, and DTTP. And so what we found was that to synthesize the A nucleotide, DATP, to synthesize thymine, you need DTTP. To synthesize guanine, you need DGTP. To synthesize cytosine, you need DCTP. And so it goes down, and do you mind if I show them that? Okay. So if you go down, I just like the way that Kristen wrote it out more. But if you go down and you have the strand here, what's in the box here is from the primer, and then everything after that is the synthesized strand. And so because of the parts that are missing, we can only synthesize the A when we're missing the DCTP. And so that's the answer to B. We can only synthesize the A and the C if we're missing the DTTP, and we can only synthesize the first 3 ATP or ACT if we are missing the DGTP. And so because that's what we were missing on the 3 parts of that question, that's how we got the answer that we did.
Okay. And then for part 5, it says, part A, it says, which of the above proteins is found in prokaryotes? And it gives you a huge list of proteins, I'm sorry. So for part A, we put the TUS, which is going to be in the termination, DNA A, DNA primase, DNA polymerase 1, DNA gyrase, and DNA polymerase 3, the alpha subunit. And then for part B, it says which of the following above proteins is found in a mouse, which is going to be eukaryotic. So we're going to find the CDC6, DNA polymerase epsilon, DNA primase, DNA polymerase 1, MCM helicase, DNA polymerase 3 alpha, and telomerase, and also pCNA. And then for part C, it asks which is involved in chromosome compaction, and we thought that telomerase and the TUS would be part of that. Part D, which of the above is involved in the initiation stage of DNA replication? We put DNA A, CDC6, DNA polymerase epsilon, DNA primase, MCM helicase, DNA polymerase alpha, and DNA polymerase 3 alpha subunit, and the MCNA. And for the last question, it asks which of the above proteins is involved in the termination stage. So we put TUS and the MCM helicase.
And that's everything. Hopefully you guys learned something, and our answers were clear. And this is our video. Enjoy.